بونجور، أهلا وسهلا فيكم بتاني سيشن من المؤتمر العلمي Research Without Boundaries. رح بلش بإنترو صغيرة عن الكلاب تبعنا. أنا اسمي آية بشر، فايس بريزيدنت لنادي بايوس أمضة. مين هو بايوس أمضة؟ هو نادي البيولوجي بكلية العلوم الجامعة اللبنانية الحدث. تأسس سنة 2019 والهدف نكون عم نساعد الطلاب ونعرفهم أكتر عن الاختصاص عن طريق اورينتيشنز السنوية اللي بنكون عم نعملهم، كمان نضوي أكتر على المجالات المتاحة لهيدا الاختصاص. كمان أيضا من أهداف النادي نكون عم نعمل أنشطة ضمن كليتنا أو بالتعاون مع كليات تانية أو حتى جامعات تانية، مثل مؤتمرات أو حتى ورش بتساعد الطلاب بعدين بسوق العمل، مثل طريقة كتابة السي في، موتيفيشن لتر، أو كيف بنستعمل لينكد إن. وكمان أيضا المشاركة بالمهرجان العلمي الكبير اللي هو بيكون أكبر نشاط خلال السنة، اللي عن جد عن طريق مختلف السكشنز قدرنا نفرجي مختلف مجالات البيولوجيا. وأخيرا أكيد ما حنطول، ما تنسوا تتابعونا على الإنستغرام، فيسبوك، لينكد إن، واتساب، حتى توصلكم كل الأنشطة والفرص المتوفرة لكم. بالنسبة للإيفنت اليوم هو اليوم الثاني من المؤتمر العلمي، وتاني اللي رح يمتد لحد نهار الأحد الأسبوع الجاي. وإن شاء الله كل يوم حنكون عم نبعت السكادجول ورح نتناول فيه مختلف العناوين مع سبيكرز كانوا طلاب جامعة لبنانية قدروا يتخطوا كل التحديات والصعوبات وحتى يوصلوا يعملوا publications أو يكملوا PhD. رح نبلش اليوم تاني سيشن مع السبيكر علي حيدر. هو a PhD student in mRNA regulation and development بجامعة مونبلييه فرنسا. راح يكون عم يحكينا أكتر عن التوبيك Spatial Organization of Translation and Translational Repression in Two Phases of Germ Granules. أهلا وسهلا علي، تفضل.
Thank you for the introduction ويعطيكم ألف عافية for the amazing work و thank you for the invitation. كمان thank you للسبيكرز اللي كانوا قبل واللي راح يجوا بعدين. It was very beautiful to all the nice talks و looking forward لكل شيء حيجي بعدين. مثل أي online session I'm gonna try to share my screen. بتصور قادرين تشوفوني؟
Yes، ماشي الحال.
Okay, cool. أكيد at any point if you have an urgent question أو حابين تسألوا أي شيء توقفوني, please feel free. kind of informal. So again اسمي علي حيدر. I'm a third year PhD student ب lab of Martin Simonelik بمونبلييه بال IGH Institute of Human Genetics بمونبلييه. اليوم حاب أحكي لكم عن research اللي أنا عم بعمله over the course of 3 years titled Spatial Organization of Translation in Two Phases of Germ Granules. Complex title بس. لما جيت لبلش لما I got the invitation I was like this isn't specific a lot, so how can I start this? وحسيت امبارح كمان علي كان عنده نفس الفكرة. فقلت اوكي حاعطي a quick overview عن how I got here, بعدين we'll talk a bit science, بعدين maybe my impression on a PhD and some points that I think are important with my little experience in the field. So I started مثل الأغلبية هون بالجامعة اللبنانية بال 2016 بالفرع الخامس بالنبطية doing biology. It was a good first year. It was an experience. We did the concours مثل كمان كتير منا. آخر السنة كمان that was an experience. بس ما توفقنا فيه. بس during the course of this year I got more sure no, I like biology. I like what I'm doing. I'm more curious about this field. فكرمال هيك كملت فيه لهذا الكورس. I took my BS وبعدين I started looking for a master's. بس بلبنان it's a bit tricky to get into research لأنه we don't have the funding to it. How it would be complicated or we need some sort of high connections to get access to actual experimental equipment. So to try إنه أعوض هذا الموضوع I tried doing 2 masters at the same time for master 1 at least to try 2 programs. أول واحد كان cell physiology and biology again بالفرع الخامس بالنبطية. Alongside with the neuroimaging program, first لأنه I liked imaging, second لأنه it was over the course of the weekend, so I could fit my programs together. Finishing the master one, this was the phase اللي بلشت فيها إنه okay, understanding what research is, how to try and think about problems, getting interested بtopics معينين ما بدي أقول hating بس getting away from other topics, so actually starting to understand what you want to do during this phase. So يمكن هيدي اللحظة اللي صرنا إنه شوي focused on what we want to do. So during the course of this year صرت أقدم على different master's programs ب fields that I felt that I like. من هود الفيلدز كانوا الجينومكس ماسترز بالجامعة اللبنانية و different programs بألمانيا وبفرنسا أو بيوروب in general. After the results I chose to continue بالcancer biology masters بجامعة مونبلييه. Especially during this period كان في البانديميك كوفيد، كان في الفاينانشال كرايسيس بلبنان كان بلشت تحتد. So it was seen إن الريسيرش بلبنان رغم إنه كان ضعيف بعد حيتأثر شوي. So I took advantage إنه I got the M2 CERIC scholarship given by University of Montpellier, which you can keep in mind, to finish my master's degree in cancer biology. Now, part of the master's program was to do an internship. I looked for several labs, but even though can my master's in cancer, the topic that I chose that really interested me, that really grabbed my attention, wasn't that much into cancer. It was more into a field شوي recent, شوي upcoming ما صار له زمان, only in 2009, which has to do with phase separation. So this interested me. I talked شوي مع responsibles تبع الماسترز, اتفقنا انه I can do my internship in this بالlab of Martin Simone, which was an amazing place to start officially my research career. So after that, during this internship, I got to know firsthand with actually trying and failing and learning new stuff on a daily basis, what research is, what kind of challenges I expect to face إذا بدي أعمل a PhD, which is a tough decision to make. Do I want to make a PhD or do I need to make a PhD لبعدين? So during this internship, I was like, I think I want to make a PhD. I want to stay in academia. I like this. So I started applying to different PhD programs, on subjects that I connected with, that I felt that I wanted to work on, which is an important part of how to choose a PhD. يمكن أحكي عنه بالآخر شوي. فبارت من هودي كان different places in Europe and the US. ومنهم كان الإيكول دكتورال بكذا محل بفرنسا. During the process, I got the CBS2 scholarship تبع الإيكول دكتورال تبع مونبلييه اللي كنت مقدم عليها مع اللاب تبعي. I got that, so I had the chance إنه أكمل for these 3 years. This year I got the grant for the 4th year لحتى to finalize my research اللي هلا حاحكي عنه شوي. باللاب in Martin's lab نحن interested in mRNA regulation and development. So the localization of mRNA, their translation, how they come together, how they get controlled. لأنه personally لما بلشنا نتعلم عن السيل خصوصي بالمدرسة كانت براسي هي مثل مسبح كبير فيها هول 3-4 organelles, nucleus, ER, Golgi, whatever. بس the more you go into it, the more you know that there are tons of details and tons of molecules inside. إذا بدنا to make it a comic, this is how to imagine a cell شوي aesthetically. But if we are to make a simulation لكمية المولكيولز اللي موجودين جوا, their kinetics, how they move, we get something like this. So for us, this was really interesting to know, if this huge amount of molecules which are present in this very small space are actually able to find themselves to do all the reactions possible in the correct place at the right time. The cell coordinates this basically through two main ways to organize this. أول وحدة هن الأورجانيز اللي هلا حكيت عنهم اللي هن عندهم membrane. بس كمان الريسنت finding كان انه في another sort of organization جوات هذا السيل اللي هو كان non-membranous organelles او phase separated condensates او RNA granules, call them what you want. بس بالنهاية هن مجموعة molecules RNA RNA and or RNA binding proteins اللي هن at a certain concentration, when their concentration reaches a certain level, they demix from their surrounding. They come together, they stop mixing مع كل شيء حواليهم, and they just form this kind of organelle. بفوتوا عليها الاشياء اللي هن بدهم اياها. They increase the reactions between the components of these RNA granules, thus allowing a more efficient function of their components. With this process, it's called phase separation. It was first discovered in C. elegans in 2009 for P bodies. But then we found that actually RNA granules are widespread. في عنا جزء كبير منهم nuclear, والinfamous اللي أكتر شيء بنعرفه ويمكن أكتر شيء بنفهمه هو النيوكليولوس. وفي different RNA granules اللي هني cytoplasmic مثل البي بوديز، في الستريس جرانيولز، وفي الجيرم جرانيولز اللي انا بشتغل عليهم. Now these RNA granules were described to be hubs of gene regulation, بس aside from the nucleolus ما كان عندنا كتير understanding of what they do and how they actually do it. Now with the increasing advancements in imaging, إذا بنطلع in details on these different granules, we could see انه they don't have this just one big macromolecule. مش كل شيء جواتهم بيت ببعضه, they form just one phase, but actually they exhibit these different phases. Aside from the nucleolus اللي we know انه it has 3 different phases مثل هود الراشن dolls اللي كان عندنا منهم ببيوتهم كانوا جوات بعضهم. We know انه النيوكليولس هود ال 3 phases are important for ribosome biogenesis. Meanwhile, for the different RNA granules غير النيوكليولس, we don't know if these different phases are functional or what do they do. So for us, this raised the question of, does the organization of RNA granules actually play a functional role? This is the big question in the phase separation field, because we're finding organelles, non-membranous condensates, but what do they do? So in the lab, we want to tackle this question based on some data we have. And so we chose the germ granules in the Drosophila as a model system, as a tool to try to understand this question. Now, ليش germ granules مش different cytoplasmic organelles? لأنه they are the best understood types of condensates in the cytoplasm regarding their biological functions. We know a lot about them, so we can help unravel the connection between the structure and function. Now, in the Drosophila embryo, I hope you can see my mouse. Germ granules are formed during oogenesis, during the end of oogenesis, whereby we have this oocyte and some mRNA called Oscar بتروح على posterior pole, and it gets translated there. Oscar nucleates these germ granules or polar granules, different name. ببلش يطلعوا عنا. So we have these germ granules at the posterior poles during the late oogenesis. Now, what happens also is that many mRNAs get localized to these germ granules. بس من وين بيجوا هول mRNAs؟ Actually بيجوا من nerve cells اللي هن, as their name suggests, might take care of the oocyte. But at the end of oogenesis, their job is over. They dump all their content into the oocyte, including هول الcontent هن maternal mRNAs. وجزء كبير منهم لهول maternal mRNAs they get localized to the germ. Now fertilization takes place. We have this new embryo. But what's interesting about the embryo of the Drosophila, especially during the first 2 hours, is that it's a transcriptionally silent sensation. يعني هو one cell في كذا nuclei بس ما في transcription yet عنه. So من وين تجي كل هيدا الريجوليشن كيف ببلش development during this stage كله بيكون تحت regulation of هود mRNAs اللي كبوهم nerve cells. These maternally inherited mRNAs. We know that germ cell formation هو part من هذا development, فهو كمان under the regulation of these maternally inherited mRNAs who are very tied to the polar granules. Now, more about these germ granules. They are made of four main components اللي هن nucleator أسكر اللي هلا حكينا عنه اللي بلاو we don't have germ granules. في the RNA helicase vasa في tudor with its many binding domains different proteins. في the PW protein aubergine اللي كنا نعرف إنه هو involved in piRNA biogenesis. More than 200 different types of mRNAs are localized بهيدي السمول سبيس اللي بنسميها الجيرم بلازما مطرح ما موجود عندنا الجيرم granules. What's interesting كمان about them is that they are organized on different levels. So first structurally إذا نطلع عليهم through electron microscopy بنشوف إنه they have this donut-like structure. We like to call it a donut. They have this electron dense shell with more loose. بس أكيد just to paint in 2D فهني more like a sphere فاضية من النص. كمان these mRNAs, these many mRNAs which are present there, they are described to be in homotypic clusters. Homotypic clusters is to say إنه the mRNA اللي هن of the same family of the same type, they don't like to mix مع شوية عنصريين. So مثلا the three examples اللي سايكلين بي اللي هن كل حدا منهم باقي اللي مثله بيضلوا سوا و they don't like mixing with other types of mRNA. بس even though كل هود mRNAs are present من أول development أول embryogenesis, they don't get translated into proteins at the same time. For example, nanose gets translated from the beginning, followed by GCL, cyclin B doesn't get translated, but actually gets repressed during the time period that we are looking at. For us, this raised the question of how do these germ granules achieve at the same time this activation repression machinery? كيف قادرين يعملوا activation و repression للmRNAs مع انهم موجودين بنفس المحل? What's the mechanism? Does the structure play a role in this? So thinking about this, رجعنا ل recent finding in the lab where we saw that actually translation activators are surrounding these germ granules. So for us, this was like maybe translation occurs at the granule surface. So we thought a bit. We came up with our working model, with our hypothesis that germ granules have some sort of functional organization. They have this biphase, a shell in the core. قلنا إنه the shell مطرح ما شفنا الactivators, it could be involved in translational activation, while the core من جوا it would be involved in translation repressors. نتوقع نلاقي translation repressors جوا أو شو we look for. كل هيدا لحتى to try to answer the question of how does it do this activation repression, and does it have anything to do with the organization of these granules? Now this model suggests إنه في عنا heterogeneous distribution للبروتينز تبع الجيرم granules لحتى يعطونا هيدي الباي phase. To check the localization of the different proteins اللي هن Oscar, Valtudor and aubergine, we wanted to use super resolution microscopy اللي هو STED نحن بحالتنا. Usually the most mainstream microscope used هو الconfocal, which is an amazing tool. But the thing with the confocal is that it excites the fluorophores of the proteins that are tagged لنا. We get this signal and we have this big signal, بيعطينا a resolution of around 200 nanometers. But what's interesting with the STED that we're using, which is a stimulated emission بس في كمان depletion of the laser, is that صح نعمل excitation للسيجنال مثل confocal بس في كمان depletion laser بهذا الشكل بيصغر لنا السيجنال اللي بيصغر عدد الفلورفورز اللي عم يقدروا يعطوا لنا سيجنال، فهيك نحصل على سيجنال اصغر more concise وبيعطينا resolution اعلى، ممكن توصل around 60 to 70 nanometers in vivo, which is huge compared to confocal او حتى different types of super resolution microscopes. So we were excited about having this. We used STED and we went to look about the localization of the different proteins. وقدرنا نشوف انه actually aubergine, Oscar, Vaza, and Tudor, they are all enriched in this shell. So what we saw in the electron microscopy was actually all these core proteins or these main proteins, let's call them, of germ granules enriched in this shell phase. As we look at the different profile plots, we see that they are actually co-localizing with each other. So using STED, we got this structure of germ granules that they have a biphasic structure with the main components enriched in the shell. So now we have the structure of germ granules. We wanted to look into translation, but we actually wanted to see translation with our eyes. لأنه I like this phrase. Seeing is believing. So, and I like imaging. So let's look at translation. And to actually see translation, we used the كمان شوي recent tool which is called SunTag. To put it very simply, for example, we have this mRNA called nanos that we want to see its translation. بدنا نشوف وين وامتى هو عم يعطينا البروتين تبعه. So what we did is that we inserted this array اسمها SunTag and it repeats. What happens is لما they get translated لما يطلع عنا هودي البيبتايدز اللي حنسميهم SunTag, they attract هيك nanobodies اللي هن short chain إذا بدكم antibodies بس صغار. They are coupled to GFP. ونحن كنا عاملينهم internal expression. So لما ينعمل translation للريبورتر mRNA اللي احنا بدنا نشوفه كانوا يعملوا attraction لهودي النانوبوديز مع GFP. So بيطلع عنا GFP signal. هيدي ال GFP signal corresponds للنيو proteins للنascent protein اللي هلا عم يطلع. We can combine this مع single molecule fish SM fish لحتى نقدر نشوف mRNA. So at the same time لما نقدر نشوف ال GFP signal corresponding to the new protein مع ال SM fish signal corresponding لل mRNA, فينا نقول إنه this is translation that is actually taking place now at this point. And so we had the tools in our hand validated to look at where translation is taking place بالgerm granule with respect to these different phases لنشوف إذا في علاقة بيناتهم أو لا. We went back to STED for the and what we saw was beautiful. لأنه we could see إنه new proteins SunTag signal ل GFP was actually mostly enriched بالshell والimmediate periphery مثل ما كنا متوقعين بالhypothesis surrounding these germ granules. في شوي but very very low amount كانت بالcore أو towards the core but not inside. We wanted to look further in more details about this. فاستعملنا 3 steps. A bit tricky بس we were looking at the germ granule. كنا عم نشوف وين موجود ال mRNA. كنا عم نشوف إذا هذا ال mRNA at the same time is being translated. What we could see is بالlower panel is لما يكون عندنا ال mRNA موجود جوات الكور we couldn't see it getting translated. بس لما هيدا ال mRNA يكون موجود بالشل أو immediate periphery, there seems to be a 50-50 chance of being translated. So for us, this place was permissive for translation or primed maybe for translation, something we want to look into. This was very interesting for us. So the localization of mRNAs with respect to their translation status could be interesting, could have an impact. So we wanted to take advantage من system at our hands اللي هو انه مثل ما قلنا قبل شوي الmRNAs صح هن موجودين كلهم من الاول بس they get translated sequentially. اول شيء nanus gets translated من الاول بعدين GCL وcyclin B is repressed, it doesn't get translated. We took advantage of this وقلنا okay, we will look at 2 time points. Early before مثلا GCL gets translated ولate. Heck, we can see a comparison, we can compare localization of the mRNAs. We categorize them in the core, in the shell, in the outside, the immediate periphery. So we started with nanus اللي we translated since the beginning. Looking at the localization of these mRNA clusters, شفنا انه nanus is mostly present in the shell and in the immediate periphery, between the air and the later phases, considering إنه nanus is always translating. This was cool with us. بعدين طلعنا on the opposite hand, which is cyclin B, which is repressed. And you could see was very interesting is that the tendency for cyclin B, which is repressed, is to be towards the core, towards the inside of germ granules, again with no difference between early and late, which fit nicely with the translation status of cyclin B. But when we looked at GCL, it was amazing when I was doing quantifications, because in the later stages we can see that GCL is more present towards the outside of these germ granules compared to earlier stages where it would be towards in the later stages it's more outside compared to earlier stages. But for us, this showed that the distribution of these mRNA clusters correlated with their translational timing. This was very interesting for us. We wanted to look at the localization of the different activators. And we saw that actually activators are كمان present in the shell, in the immediate periphery, co-localizing with the proteins of germ granules. But this was very nice. Our hypothesis was going very nicely. We were guessing it all. So we're like, okay, let's look at repressors. أكيد they are present on the inside, repressing mRNAs اللي جوا. But what you could see is, looking at this a really long list of repressors, we couldn't find ولا واحد present in the core. We couldn't find ولا واحد actually inside repressing. So we're thinking, okay, there should be some other way of repression. But we'll keep that in mind. There could be some other sort of way to repress these mRNAs. Well, now, okay, maybe it has to do with the compaction, with the orientation of these mRNAs. So what we did is that رجعنا للSM fish, the single molecule fish, we tagged the 5' end of the mRNAs with 3' end of the mRNAs لحتى نشوف how they are organized. We took the two extremes of the spectrum. أخذنا نانوس اللي هي دايما translating، أخذنا cyclin B اللي هي دايما repressed. We're looking at translating nanos, we found a really beautiful picture which was like الوردة الروزات اللي هي إنه شفنا إنه 5' of these mRNAs is present more towards the outside, towards the shell, while the 3' of these translating nanos mRNAs is mostly present towards the core of germ granules. We're like, okay, let's look at cyclin B, more compact. But what we saw is that looking at the 5' and the 3' was we had an inversion of this conformation. شفنا العكس إنه cyclin B, which is being repressed, has this different orientation, while 5' is hidden in the core, while the prime is more towards the outside. This was quite interesting. Okay, قلنا بدنا نعمل validation, some way to sort of approach to validate this localization of mRNA clusters, this orientation. إذا نخلي mRNA ينعمل له repression، هل حيقلب مثل cyclin B؟ هل حيصير له شيء ثاني؟ So we wanted to validate this. ونعمل هيك we used a pengu mutant، بس اسمه pengu دايما بتذكر this little guy. So whenever you see him واقف بالزاوية يكون بهيدا الكونتكست. So in the Pengu mutant بيصير في عنا Pengu is a kinase involved in the phosphorylation of several repressors during the activation of the oocyte just before fertilization, which leads to a global transcriptome remodeling. We combine this genetic background مع Santec tool وشفنا إنه in this background we have a decrease in translation. We're like, this is very cool. We have a condition where we have less translation. Let's see how that impacts the mRNAs which are present there. So رجعنا ل two extremes, nanos translating وcycling B لحتى نشوف does their localization or orientation change. شفنا nanos بالأول شفنا إنه what we could see is that in the Pengu mutant compared to wild type, it's more present towards the core, which correlates with its with less translation of nanos. While for our control, which was cyclin B, which wasn't translating in both cases, we still had it present more towards the inside. And okay, this is nice, but what about the orientation? We expected to see in nanos with the 5' برا with 3' جوا. In this context, هيقلب هيصير مثل cyclin B وهيبرمها. But what we could see is that it didn't change its orientation, but looking at the images, we saw that the 5' and 3', they look closer. They seem to be more compact on each other. Like, okay, let's quantify this. Let's see if this is real or not. أول شيء we measured the precision تبع our system, and we could see that we can measure distances أعلى من 27 nanometers. So let's see if the distances between them is more than that, and we can check that. We checked for wild type distances between 5' and 3' of translating nanos, and we can see it was about 150, 160 nanometers. But لما طلعنا in the Pengu mutant with less translation of nanos, we could see is that the 5' and 3' are actually more closer to each other. The mRNA is more compact on each other with the median distance of around 100 nanometers. We looked for cyclin B, which is repressed, to see what's also going on. And صح ما في فرق بين Pengu والwild type, which was expected because they behave the same, cyclin B in these cases. But it's repressed, and what was very cool is that it's repressed and the mRNA are more compact on each other. So this led us to think إنه صح ما في عنا translation repressors in the core or in the germ granules per se. But the other mechanism that we're trying to test is that the repressed mRNAs could be just repressed because they are more compact than the translating mRNAs. So far, we can see that we have different aspects of the effect, the translation status on the localization, on the organization, on the orientation of these different mRNAs. What happens if we actually mess up this biphase, this shell core structures? لازم نخربها إذا تروح هيصير معنا بالtranslation. For that, we took advantage of another mutant لواحد من main proteins of the germ granule حكينا عنه اللي هو جدور. جدور هو protein عنده 11 main domains involved in different bindings for different proteins. The mutant that we used اسمه جدور A36. It has a point mutation turning الارجينين into tryptophan. This point mutation بتخرب شيء اسمه aromatic cage بهذا الدومين. It could alter some aspects of the germ granules. It was described إنه mRNA present there. They localize normally to the germs. They are there. They are present. Or germ granules are present there. But the number of germ cells اللي بيطلعوا من development is much less. It's 3 compared to 24. Using electron microscopy study compared لل big donut that we used to have in mutant, they were described to be bars or rods. Seems very interesting. Let's look for us instead using fluorescent proteins. How does this look like? So رحنا للstead, and we couldn't see these bar-shaped granules, but what we could see is that we lose this donut structure, and we have these much smaller single-phase germ granules, and we're like, okay, we have a condition where we lose this biphasic structure. Does it affect translation? So we combined this tool with SunTag, and what we could see is that this translation is strongly reduced. The Hodel mutants, they lose this biphasic, suggesting that this biphasic structure could be important in the maintaining this translation present here. So رجعنا فكرنا انه this single phased smaller granules could be a consequence مش a cause of translation. So رجعنا شوي بالزمن اذا بدنا للستيج 14 oocytes قبل الامبريو، قبل fertilization، قبل كل شيء، حتى قبل ما translation للكلش، وقلنا اوكي اذا هن فعليا consequence فمن الاول لازم يكونوا بالاول لازم يكونوا donut shaped، لازم يكونوا round biphasic، ولانه في عندنا مشكله بالترانسليشن عم بيسكروا على حالهم. But what we could see is that even in stage 14 oocytes, even before translation started, these germ granules are small, are compact, and they don't have this biphasic structure, suggesting that indeed the structure affects the translation. With that, I want to sum up. The points that I tried to show you was where the germ granule functions are compartmentalized, the biphasic shell core structure. No translation takes place in the shell. The 5' of these mRNAs is more towards the outside, the 3' more hidden. For repressed mRNAs, the 5' is more inside, while the 3' is more towards the exterior. That repression depends on the compaction level of these mRNAs. When losing the germ granule biphase affects the translation. Putting all these experiments together with a few others with many controls and many confirmations, we were able to put this into a very beautiful paper. This was the work of myself and Anne Ramat, a researcher in our lab, and you can find the paper on bioRxiv for now, hopefully soon in publication. And with that, I would like to thank you with just one more point to add. This was an amazing experience for me, and if I am to reflect on it going back, my main question would be, do I want to do a PhD or not? What I saw is that doing a PhD, it's not just شهادة زيادة طلع على الhead. It's nice to have it, but مثل أي شيء تاني, it has its positives and it has its negatives. It has its lovely moments, it has its struggles. That أي حدا عم بفكر to do it just needs to and to actually be sure that they want to do it, لأنه it's an amazing experience. It gives you a really good push. But if this push is in the direction you want to pursue, يعني really think about what you want to do in the future. لأنه many of the struggles in the PhD, you would have much less time to interact. You would have deadlines. You would have ضغط. You would sometimes have to leave your family, leave your friends, live in a new place, integrate with new societies. This could be difficult, but at the same time, you would be following something that you love. You would be doing something new, meeting new people, understanding different cultures, communicating science, which I think هو الشيء اللي جمعنا اليوم throughout this conference, which is our love for science and our love to share and exchange the different scientific values, our discoveries, how we think about science. Because the more we in this field, this هذا المجال, the more we learn stuff, the more we change our way of thinking. We're definitely not the same as لما كنا بأول سنة جامعة. We're definitely thinking about science much more nicely, much more beautifully, much more sophisticated. So this is something to keep in mind. Maybe a couple of things to mention was that part من ال PhD, which is I love giving talks, I love talking about science, I love communicating. Part of my experience was being part of the 3-minute thesis in which is like a competition where you prepare a speech, a public audience, to talk about your science, which is something nice. لأن دايما ممكن حدا من برا يسألنا طب أنتوا شو عم تعملوا؟ We would be studying something very specific and we're like والله مرة فكرت كيف بقدر أشرحها للعالم. So this was a really nice experience. I mean, my set is on YouTube if you want to check it. But like we would like to make science more accessible for the public. Part of this, my last slide would be, this is something that I'm working on with Fadl Faqih. He's a PhD student in Czech Republic in Lukish Lab. بتصور هو معنا كمان. His credentials are here. We started this blog لأنه أنا وياه we're enthusiasts, let's say, about the RNA and RNA stuff. So we called our blog rnastuff.com. وجواتها we try to write articles about the different advancements in science, especially in RNA, different experiences, some philosophy of science. If you have a look, when you have some time, reach out to us if you would like to share something, to write about something. Please, we're open to that. Maybe last two things would be a thank you to my lab, especially my team for giving me the opportunity, for Anne for the huge work, and Celine for the help, our collaborators, the platforms that we use, the funding, and just be curious, study well. It's not the most important thing, but it is important. And have some good connections with people. Be curious, always follow the new stuff, and follow the science rather than following just the people. Or follow your heart that follows the science. With that, again, sorry طولت عليكم, but I will end with that. Thank you so much. Please, if you need to talk to me, I have you have my credentials, my contact info on X, on my email or LinkedIn, whatever you guys want. And thank you. I'd be happy to answer any question that you have. Please, Ali.
I want to spoil the fun هيك بشوية questions إذا ما في مشكلة.
Please.
So I want to start في كذا سؤال بس يمكن بلش من الأساس من البداية. أنت you mentioned that you did your masters in neuroimaging وعملتها كمان بالنبطية cell biology إذا مش غلطان. To what extent, clearly you are using this neuroimaging background that you had in your research today, but to what extent was it actually useful? Reflecting on it, was it worth spending your weekends doing a second master's, or would it be maybe better to do either something else or, focus on different projects?
If I am to give an advice, if we have the opportunity to do an internship, for me, that would top any master's or any actual just theoretical stuff. لأنه being inside the lab, being with actual contact with science is what starts to change the way we think, is what starts to give us new ideas. It's what really puts what we know into action. So I say if we have the opportunity, or what we should look for to try and find an internship rather than doing another master's, or just increasing the education part بالCV rather than increasing the experience part. Unless we don't have the choice, then we need to try to increase better our background maybe.
Thank you. بالنسبة للscientific questions, maybe going more towards that هلا. You mentioned at the beginning, sorry, completely naive about the topic. We might have some overlap, by the way, بالcyclins. We can discuss that maybe later privately. But بالنسبة للDrosophila, you mentioned that you use Drosophila as a model system. Would you tell us why exactly Drosophila system تاني استخدمته أنتم هم?
It's first it's the model system used by the lab لأنه في huge overlap with Drosophila with human genome. It's an in vivo system rather than just working with cells. So we have the whole system working. We have a more physiological context of what's going on, rather than just doing artificial granules or just putting stuff in cells and see what could that happen. That's very useful. That's and the lab more oriented towards seeing the stuff in the in vivo context rather than in cells. Kaman, what helps with Drosophila is the short development times, which is about 2 weeks. So in 2 weeks, we could have a new fly. We could see what goes on during different generations.
Lovely. Last question. I mean, clearly you have done quite a lot of research, but at least to me, it looks like this is more towards the fundamentals of science. أكتر ما هو translatable, more patient oriented. So my question would be, how can this be used as a بدك in a more translatable how can we translate the information that you to us into a more clinic setting, and how can that be useful for us in hopefully the few coming years?
هذا كان آخر question لما كنت عم defending my masters كان إنه okay, fine, this is cool, but how can we use this? Because I was doing a cancer program, and usually cancer people are more like, how can we use this, rather than بس بدي أفهم what's going on. But the thing which is beautiful about fundamental science, which I love, is that when you understand how stuff works, you can definitely find some way to use. And the new thing in the phase separation field, or in the granules field, is that we know that they are implicated in the controls of different functions, different mechanisms through a cell. So what's currently what's currently being tried to be done is using these germ granules to actually control translation, to control regulation, playing on the physiological properties of this. Mainly, it would be in the neurodegenerative fields, because as we get older, as we have more neurodegeneration, we have the formation of these solid granules with different physiological properties that would alter and affect different mechanisms and the functions that they also have. So understanding how these granules actually form, how they work, how their biophysical properties are affected, would help us solve these kind of problems.
Very cool. Thank you.
Thank you.
بعد حدا عنده أي question؟
تفضل بليز.
مرحبا. بدي اتابع شوي على اللي قالوا عليه، اذا لان شوي استفزني السؤال انه هل ضروري عن جد يكون كل ساينس احنا عم نعمله يكون له implementation in medicine and translational research, or we just do science for fun? شو رأي علي بالموضوع؟
هلا لحظة، أي علي منه عشان
I think
هلا فيهم الاثنين يجاوبوا، ما في مشكلة.
بتصور بلش انت.
Okay, sure. هلا I mentioned in my talk yesterday إن أنا كمان بشتغل pure fundamental science with protein actors and the likes. I think I do agree with the whole point about does everything need to have something that is more towards translatable medicine, something that is more patient oriented. Yes and no. يعني نحن ما فيناش نكون بس عم ندرس شغلة that is extremely arbitrary to a point that the funding could be otherwise used in something that is more كيف بدي أقول urgent. عرفت كيف؟ يعني أنا مثلا اليوم ما فيني أروح أدرس disease هيدا ببين in one patient out of every 5, 6, 10 million، وآخر شيء يكون في عندي something that is as frequent as Alzheimer's or ALS or otherwise. I'm not saying that we should completely stop the research on the other topics. What I'm trying to say is that the priorities should be on something that is more urgent given the circumstances right now. يعني أنا مش اليوم مثلا يكون عندي patient that is presented مثلا امبارح I presented in my data that في عندنا mutations بالبروتين اللي أنا بشتغل عليه is up 70. However, it is not something that is Alzheimer's, right? For example, does this mean that we should stop working on that? Obviously not. But what I do mean is that if there's more work or more brains that are working on Alzheimer's, I wouldn't be particularly sad. Because when it comes to the priorities and the funding, نحن هلا أنا مأكد أنت فضل بتشتغل بالتشيك، علي بيشتغل بفرنسا وهون بألمانيا. The funding, at least from my viewpoint, when the بقولوا Germany is, you know, the funding father ومش عارف شو of science in Europe, it's going down the drain. At least this is the way I'm seeing it from the funding institutions هون. فإذا نحن عم نشوف إنه الفunding عم بقل، ونحن أساساً تركيزنا عم بكون more towards niche, extremely niche topics. هيدا ممكن يكون عم بيسبب لنا مشاكل for later. long story short, ضروري نحن نكون عم نكفي fundamental research مثل نحن بعد 3 بس كمان بنفس الوقت هيدا الشيء ما بيعني انه نحن بدنا نكون عم نحط priority على هدول القصص اكثر من الاشياء يلي اذا بدكم more urgent currently.
To reflect on that, the sort of fundamental research is a bit overlooked بالموضوع. بس يمكن من بعيد شوي survival of the species, we would like to let's keep it going or let the weak be gone. But from a science point of view, as a scientist, it's too big to say I'm a scientist, but as somebody that works in science, part of the enjoyment that we have, part of the enthusiasm for science, he understanding how this universe works. And I think we can always get stuff from كل شيء that we learn. يمكن المشكلة هي فهمان قصدك ويمكن أنا فاضل تناقشين كتير بموضوع إنه where should the funding go يمكن تروح rather than let's give everybody more money.
Thank you, Ali. I'm just curious about Fadl's response also to that, because yeah, I'm really curious.
لا مش خلينا نقول it's a fine line يعني بين انه تحط بس تدرس اشياء خاصه بال fundamental stuff او انه تلاقي اشياء translational عن جد تعمل بس I agree with علي بموضوع انه science is just to know how the universe work, how like my PI once mentioned انه في proteins ببارازايت بيعيش بالبحر does not affect us at all, بس هذا أكتر protein abundant on earth, so we just want to know why it's there, what's doing, so it's just the curiosity that fuels us.
Totally agree. في شغله بس I think one of the major breakthroughs in science was إذا مش غلطان كان اسمه الTAC polymerase that is basically used everywhere right now. It is found in one of those sea creatures. I completely blank on its name. But the fact that this is the work of fundamental science. أو الأشياء that is not basically cancer research or immunology, you know.
GFP, CRISPR, كل هذول الأشياء اللي فعليا من basic research.
اوكي يمكن ما هناخد الدسكشن نحكي فيه بعدين عشان ناخد لكم كتير اكتر من وقتكم. Thank you again for the organizers, for everybody for listening.
ميرسي لك، يعطيك الف عافيه.
Thank you.